Okay, so welcome to this next video in which we are discussing the adrenoreceptors. Okay, right, so we are looking at the way by which the alpha-1 adrenoreceptor triggers the phosphorylation of the regulatory myosin light chain to trigger the activation of the sliding filament mechanism of muscle contraction and therefore trigger the contraction of the smooth muscle cell. Okay, right, so we are in the process of discussing the structure of phosphatidylinositol 4,5-bisphosphate. So, I was just talking about the optical isomer of inositol that is used in phosphatidylinositol 4,5-bisphosphate, and in fact the form of inositol that's actually used all over the place within a cell, because the other optical isomers of inositol aren't used so much in cells. So, let's have a look at this now. So, basically, what I want to show you is the structure then of myoinositol. Okay, so, myoinositol basically has 4 of the alcohol groups coming out of the page towards us, and then the other 2 going back into the page away from us. Okay, so let's show this. So, basically, what we're going to have is this alcohol group here is going to come out of the page towards us, like so. Okay, so this is coming out of the page towards us. Then this one here is also going to come out of the page towards us. Okay, like so. Then this one's going to come out of the page towards us. So remember, the reason that inositol has so many optical isomers is that the alcohol groups can either come out of the page towards us or they can go into the page away from us. Okay, so these ones are all coming out of the page towards us, and then the hydrogens that come off these carbons are going back into the page away from us. Okay, then the next alcohol group of this 4th carbon is going to go into the page away from us. The alcohol group of this 5th carbon up here is going to come out of the page towards us. Okay, and then the alcohol group of this 6th carbon over here is going to then go into the page away from us. Okay, right, so this then is the structure of myoinositol. Okay, so you have 2 on one side, 4 on the other. Okay, and then the 2 on the opposite side, those have a single alcohol group that's on the side coming out towards us between them, basically. Okay, so this is myoinositol. Now, there is a set naming system for myoinositol, which is why we're discussing this at all. Basically, the naming system is that you give this carbon here the number 1, you give this carbon here the number 2, this carbon here the number 3, this one the number 4, this one the number 5, and this one the number 6. Now, this alcohol group here is the one that's going to be bound via the phosphoester link to this phosphate group of the phosphatidate molecule. Okay, and then the rest of them are going to still be free over here. Okay, so what we have now created is phosphatidylinositol. Well, we haven't created 4,5-bisphosphate yet, but we've created the phosphatidylinositol. We have got a phosphatidate molecule linked to an inositol ring, so that's known as phosphatidylinositol, or just PI for short. Now, we want, however, phosphatidylinositol 4,5-bisphosphate, and to turn phosphatidylinositol PI into phosphatidylinositol 4,5-bisphosphate, we need to stick phosphate groups off the inositol ring. Okay, and now we know exactly which carbons we are going to stick these phosphate groups off, because I've told you the naming system for the carbons of the inositol ring. So we're going to have one coming off this 4th carbon down here, and then one coming off the 5th carbon down here. Now, this lengthy discussion of the optical isomerism of inositol was all to try and help you to understand why this is not called phosphatidylinositol 3,4-bisphosphate. Previously, before understanding that there is a specific optical isomer of inositol that we are using here. You might have been tempted to call this molecule phosphatidyl inositol, and then we'll call this carbon 3 and this carbon 4. So you might have been tempted to call it phosphatidyl inositol 3,4 bisphosphate, but it's 4,5 bisphosphate, and that's because of the specific optical isomerism of inositol that we are using. We're using specifically myoinositol, and the carbons of myoinositol are pre-named for us. Okay, right, so it's this phosphate group here, it's this alcohol group here off this 5th carbon that you've got this phosphate group being added onto rather than this one. And you might wonder, well, what's the difference? Well, if it's been added onto this one, it's next to an alcohol group that's going, well, it's between 2 alcohol groups that are going into the page, whereas if it's on this one, it's next to 1 alcohol group that's going into the page and 1 that's coming out towards us. Okay, so there is a difference, basically. Right, so that's why it's phosphatidyl inositol 4,5 bisphosphate. So this is the structure of phosphatidyl inositol 4,5 bisphosphate. And I hope what I've convinced you of is that this is just a modified phospholipid. It's just a phospholipid with an extra bit added onto it. It's got this inositol ring and then these 2 phosphate groups that dangle into the cytoplasm. So effectively it's just a phospholipid that has a bigger head, basically. Okay, right. So it is this molecule, phosphatidyl inositol 4,5 bisphosphate, which is the target for phospholipase C enzymes. Okay, so we have just activated the phospholipase C beta enzyme by the binding of the alpha-Q GTP subunit to that phospholipase C beta enzyme. Okay, and what the phospholipase C beta enzyme is now going to do is it's going to act on these phosphatidyl inositol 4,5 bisphosphate molecules, and it's going to cleave them into diacylglycerol molecules and also inositol 1,4,5 trisphosphate molecules. So basically what it's going to do is it's going to cut PIP2 molecules across here. It's going to hydrolyze the phosphoester link between the 3rd alcohol group of the glycerol molecule and the alcohol group of the phosphate group here, or rather the carboxylic acid-like group of the phosphate group here. Okay, and the 2 products you're then going to produce, if we draw these out, you're going to produce a molecule which has 2 long-chain carboxylic acids esterified to a glycerol molecule, like so, and this will remain attached to the inner leaflet of the phospholipid bilayer, so it will remain within that inner layer of phospholipids in the phospholipid bilayer. Okay, because it's still got these long-chain carboxylic acids anchoring it in. Now, what's the name for this molecule? Well, basically, this molecule is called diacyl and then glycerol. Okay, and the reason it's called that is basically it's a glycerol molecule, which is the portion in green, and now it's got 2 long-chain carboxylic acids esterified to it. Now, to understand where this acyl comes from, we need to understand what an acyl group actually is. So if we look at a carboxylic acid molecule here, so I'll put an R group there to denote it's got an arbitrary tail there. So this is a carboxylic acid molecule. Now when you esterify it to an alcohol group, what do? You remove the alcohol group from the carboxylic acid molecule, you remove the hydrogen from the alcohol group, you bind those 2 things together to produce water, and you then bind the carbon to the oxygen here to create the ester link. Okay, so actually you do not add the entire carboxylic acid molecule on. Instead, you add this portion of the carboxylic acid molecule on here. Okay, this is the acyl group of the carboxylic acid molecule. Okay, so this is the acyl group. Right, so you only add on the acyl group, you don't add on this alcohol group, so you don't actually add on the carboxylic acid, you add the acyl group of the carboxylic acid. So that's why this is called diacylglycerol, because you've effectively added on 2 two acyl groups onto a glycerol molecule, hence diacylglycerol. Now sometimes it's also called diglyceride, that's another name for this kind of a molecule, and often diacylglycerol is actually abbreviated just to DAG, D for di, A for acyl, and then G for glycerol. Okay, so that's one of the products, and that remains in the plasma membrane. Okay, the other product that you produce is the inositol molecule with the free phosphate groups bound to it. So here's our inositol ring, this six-membered ring, and now we've got phosphate groups coming off the first, the fourth, and the fifth carbons, and we know that they are the first, the fourth, and the fifth carbons, not the first, the third, and the fourth, because we know we're dealing with a specific optical isomer of inositol. Now, this molecule is called inositol, that makes sense, and then 1,4,5-trisphosphate. Now, if you were being really strict, you would actually call this myo-inositol 1,4,5-trisphosphate, i.e. you would tell me what optical isomer of inositol I was dealing with. Because we're generally dealing, because we are biologists and we are generally dealing with living systems, the only form of inositol that's physiologically relevant is myo-inositol. So whenever you say inositol, everyone knows what you mean. Basically, you mean myo-inositol. But if you were being really strict, you would put myo-inositol 1,4,5-trisphosphate. Now, inositol 1,4,5-trisphosphate is often just abbreviated to IP3. Okay, so the I for inositol, the P for phosphate, and then because you've got three phosphate groups, we'll put 3 at the subscript. Okay, so here is the inositol ring in blue. Okay, then we have phosphate groups coming off the first here, the fourth, and the fifth carbon, like so. Now, the IP3 molecules do not remain at the plasma membrane, okay? They were only attached to the plasma membrane previously because they were being held onto by the diacylglycerol molecule, but now they'll just drop off into the cytoplasm, basically. Okay, now for our purposes, we're not going to really be interested in what diacylglycerol is going to do. We're going to be very, very interested in what inositol 1,4,5-trisphosphate is going to do, because it's going to go off and act on receptors for inositol 1,4,5-trisphosphate that are within the ER membrane. So let me just discuss this. So basically, if this is our smooth muscle cell here, then there is an intracellular organelle within the smooth muscle cell called the endoplasmic reticulum. Okay, and this is a membrane-bound intracellular organelle, so it has a membrane around its entire volume. Okay, so this is the endoplasmic reticulum. Now, one of the functions of this very important intracellular organelle is that it has a large store of calcium ions within its lumen, basically. Okay, so calcium concentration within the cytoplasm of the cell is kept at extremely low levels, a level of around 100 nanomolar. And if you contrast that to the concentration outside the cell, which is around 1.5 millimolar, that is a 15,000-fold gradient of calcium across the cell membrane. So it's quite a fantastic feat on the part of the cell that it keeps calcium so low within the cytoplasm of the cell. Okay, however, calcium is one of the most important signaling molecules within cell biology. The other being, it's up there with cyclic AMP as an extremely important signaling molecule. So what you have is an intracellular store which contains a larger concentration of calcium, and you can release this calcium into the cytoplasm to trigger all sorts of responses, and it's the release of this calcium that is actually going to trigger the phosphorylation of the regulatory myosin light chain. Okay, right, so IP3 going up within the cytoplasm is going to act on proteins within the membrane of the ER and lead to the release of calcium from the ER. So let's now have a look at that. Okay, right. So let me draw the ER again. Okay, so let's say this is the membrane of the ER. So basically within the membrane of the ER, you have a very large protein, basically. Okay, and this is what's known as the IP3 receptor. Okay, like so. And basically it's a massive great complex of multiple proteins stuck together. So it's actually a tetramer, and it has 4 proteins that are all stuck together. Okay, so this complex of 4 separate proteins is called the IP3R for IP3 receptor. Okay, and it consists of 4 separate proteins stuck together. And what I want to now do is pull out one of the subunits of the IP3 receptor and have a look at its membrane spanning topology. Okay, so I want to pull it out and have a look at its structure. Okay, so if I have this membrane here, then let's put the membrane spanning topology of this protein within this membrane. Okay, and it's important to remember that this side of the membrane now represents the cytoplasm, okay, and this portion represents the lumen, okay, and similarly for this membrane, this side will represent the cytoplasm and this side will represent the lumen. So basically I'm just transferring this membrane down to here, and we're going to have a look at the more in detailed structure of what I'd just drawn as a quarter of this channel before. Okay, so basically both the amino and carboxy termini of the protein will be in the cytoplasm. So here's the amino terminus here, and it's in the cytoplasm, and then it will have a membrane spanning alpha helix, then it will have a second membrane spanning alpha helix, a third, a fourth, a fifth. You'll then try to straddle the membrane, fail, that's a P loop, then you'll go back another membrane spanning alpha helix, and then you'll finish with the carboxylic acid tail. So this is the membrane spanning topology of one of these subunits of the IP3 receptor, which I'll highlight in pink here. Okay, right. So basically you put 4 of these together to make an IP3 receptor then. Okay, and they are massive great proteins. They are around 2,750 amino acids. So these are big proteins. Okay, now the simple thing would be if there was just one gene which coded for the IP3 receptor subunit, and you just made 4 of these proteins, stuck them all together, and that was your IP3 receptor. The reality is it's a little bit more complicated than that. There are actually 3 genes for IP3 receptor subunits. Okay, there is the IP3 receptor 1, okay, subunit, the IP3 receptor 2 subunit, and the IP3 receptor 3 subunit. Okay, so there are these 3 different genes which all create different, well, they'll all have slightly different sequences of organic bases, and therefore the protein that they produce will have slightly different sequences of amino acids, and they will all have this same membrane spanning topology, and they will all be capable of aggregating together to form a tetrameric IP3 receptor, but they will produce channels with slightly different properties. So, basically the next thing to ask is how do you assemble them into tetramers? I.e., is it the case that you just take 4 IP3 receptor type 1 subunits, stick those 4 identical proteins together to produce an IP3 receptor and call that the IP3 receptor type 1? Okay? No, basically. You can do that. You can form homotetramers of the IP3 receptor subunits, but more commonly you end up with heterotetramers, okay, which is basically where you produce tetramers which have more than one of the IP3 receptor subunits within, well, more than one type of IP3 receptor subunits within them, i.e. they're built out of more than one type of IP3 receptor subunit. Okay, so you get homotetramers and heterotetramers. Okay, right. So, that's the IP3 receptor. Let's now talk about what IP3 is actually going to do to this receptor. So we've got these 4 subunits, and basically what will happen is each of these subunits has a binding site for IP3. Okay, so let me discuss this. So we'll look at the receptor from the cytoplasmic aspect. We're imagining we're a little man looking down on the receptor, and here are the 4 separate subunits out of which it's built. Okay, 1, 2, 3, 4. Now, basically, IP3 itself doesn't actually cause the receptor to open on its own. Instead, what it does is it causes a change in conformation that then leads to another substance being able to activate the opening of the IP3 receptors. Okay, so let me discuss this with you in a bit more detail. So basically, before IP3 is bound to the IP3 receptor, all 4 of these IP3 receptor subunits have on their cytoplasmic surface a calcium binding site that is inhibitory. Okay, so this is an inhibitory calcium binding site. Okay, so if calcium binds to this inhibitory calcium binding site, it will make the chance that the IP3 receptor will open even lower, basically, than it was when calcium wasn't bound. Inhibitory calcium binding site. Okay, there is then also a binding site for IP3. So each one of the receptor subunits will have a binding site for IP3. So this is the IP3 binding site. Basically, when IP3 goes up in the cytoplasm, because you have activated the phosphoinositide pathway, which is the pathway we've just discussed, so where you start with the heterotrimeric GQ pathway, which then activates the phospholipase C-beta, which breaks down PIP2 into diacylglycerol and IP3. That's what's known as the, where should I put this? This is a big title. And that's what's known as the phosphoinositide pathway. Okay, so phosphoinositide pathway. Okay, so when you activate the phosphoinositide pathway, IP3 will go up, and the IP3 will be able to bind to these IP3 binding sites on the cytoplasmic surface of the IP3 receptor. And what this triggers is it triggers a conformational change in the IP3 receptor subunits, which then leads to the removal of the inhibitory calcium binding site from being exposed and the exposure of a stimulatory calcium binding site. So let me show this. So what will happen is when IP3 binds to the IP3 receptor binding sites, okay, for IP3, then what will happen is all of these subunits will change conformation, and the inhibitory calcium binding site that was on the surface of the IP3 receptor subunits now becomes, well, it's taken away from being exposed, so it's no longer exposed. And instead what happens is you get a new binding site becoming exposed, and this is a stimulatory calcium binding site. So this new binding site here, which I'm colouring in green, this is a stimulatory calcium binding site. Okay, so now if calcium comes and binds to this stimulatory calcium binding site, then the IP3 receptor will open. So IP3 does not cause the IP3 receptor to open directly. Instead, it changes the conformation of all of the subunits so that they take away this inhibitory calcium binding site that they had and then expose this stimulatory calcium binding site. And now if calcium ions come and bind to these 4 stimulatory calcium binding sites, then that will trigger the IP3 receptor to open and release the calcium from the ER into the cytoplasm. Okay, now what I want to do next is talk about the different forms of calcium signals that you can then produce within a cell. Okay, so there are different forms that the calcium signal can take on, and I want to discuss those different forms in the next video.